Effect of photosynthesis and carbohydrate status on respiratory rates and the involvement of the alternative pathway in leaf respiration. You may be thinking, what does this title even mean and what is some background of this? This title for this article is how photosynthesis and amounts of carbs can affect respiration and how the alternative pathway is involved in respiration. Carbs are necessary in plants to carry out various things such as growth and cell protection. Photosynthesis and amount of carbs can easily affect how respiration works, and this lab goes way beyond that, just by mentioning the alternative pathway, which many don't know about. The alternative pathway in plants refers to a non-energy terminal in the mitochondrial electron transport chain. This is used to relax the coupling electron transport reactions leading to metabolic homeostasis. So the hypotheses and predictions given are that there is a direct relationship between respiration rates and the carb status in plant tissues, as well as the investigate demonstrates the relationship between respiratory O2 uptakes in the dark and the involvement of the alternative path and carb levels in the leaves of wheat and other species. They also showed to the degree of which the alternative pathway participates in normal respiration and how it's directly related to the level of sugars in the leaf. So covering the basics of the experiment, the main materials used are common wheat, common pea, spinach, a leaf area meter to get the O2 uptake rates, an electrode cuvette with 105 milliliters of reaction medium, 2 razor blades, a nylon net, IR gas analyzer, and an O2 electrode. So for a quick side note, the method of this experiment is very long and confusing, so if it's easier to just pause here and read the method on this slide and the next slide, it may be a lot more clear, but I will go through some of it. So a major part of this experiment is taking these leaves, doing these various things with them, measuring the uptake level, see if there's any change, and then redoing that until they see a specific change. So this is why going through this would be a really long process and take a ridiculous amount of minutes to go through, but I am including every step on here. So the data does show that uptake rates in spinach and wheat leaves in the dark were faster after the plants were allowed a specific period of photosynthesis for several hours, and the alternative pathway did increase after the period of photosynthesis. The rate of O2 uptake by wheat leaves in the dark was lower in leaves harvested at the end of the day versus leaves harvested after periods of photosynthesis. And for pea leaves, the total carbs decreased and the levels of glucose and fructose slightly increased overnight, but no decrease in respiratory rates. So this is the graph constructed by me. This shows that the casein concentration with O2 uptake of wheat leaves and sham, which is one of the solutions, is in the blue dots, and without sham is the little purple flowers. Now this table shows the oxygen uptakes by mitochondria isolated, so this has to do with the alternative pathways, and this shows each leaf that was tested. So discussion, was the hypothesis and prediction correct? Yes, the hypothesis is correct, as there is a correlation of the alternative paths, sugars in the plants, and the O2 uptake. Although that was proven, they are still left with the function of the alternative pathway being unknown, because there is very little actually done on that part. Going back to the data, the first graph shows that the alternative pathway in wheat leaves had 80% of the total amount of respiration in leaves harvested after a specific period in the light. And it is concluded that the level of respiration in leaves determines the O2 uptake and how the alternative pathway contributes to the uptake. This study was a very long and difficult one to conduct, as there were multiple portions, different charts, data, graphs, of various things, because you're testing 3 different kinds of species of plants. But the main focus was about how photosynthesis can change these rates of O2 uptakes in various species, different lightings, and even different temperatures. But this can be furthered into studies of alternative pathways and respiration. What are they used for? What's the purpose of these different pathways? What causes them to kick in? And there actually isn't a lot of studies done for these pathways, which makes this an even more interesting study that can be turned into many other different kinds of studies. So my concluding remarks are thank you for watching, thank you for your time, and I really hope you enjoyed this.